everyone, so today we're going to be talking about biological molecules. So the key points that we're going to be discussing is the building blocks of life, monomers, polymers, and macromolecules, carbohydrates, dipoles, and hydrogen bond, lipids, proteins, and water. So we'll first start with the building blocks of life. This is very simple. Basically, the monosaccharides, amino acid, fatty acid, and glycerol, organic base, and monosaccharide. All these three are basically the molecules that will make up the bigger molecules. As you can see in the image here. So you can see here, monosaccharide will make up to polysaccharide. Organic bases with monosaccharide will make up nucleotides, which then will become nucleic acid. Amino acid will become proteins, and fatty acid and glycerol will become lipids. So these are the basic key points that makes up our biological molecules. So next we're going to be looking into monomer, polymer, and macromolecule. So this is basically how the process of macromolecule, monomer, and polymer goes. So it will first start with a macromolecule. Macromolecule is a giant molecule of proteins and nucleic acid, which each of this macromolecule will form together and become a monomer. Monomer is a basic building block for the synthesis of a polymer. So many of the monomers will join together to make polymer. Polymer, the word poly comes from the derived meaning called many. So poly means many. That's why it's polymer instead of monomer. Monomer is one, polymer is many. So polymer are giant molecule made up of many similar repeating units joined together in a chain. So the examples of polymer are polysaccharides, polypeptides are for proteins, and polynucleotides are for nucleic acid. So next, we will look into carbohydrates. So these are the key points that you will need to know.
To make up the carbohydrates, we only need C, H, and O. So carbon, hydrogen, and oxygen. The ratio of these three is 1 from carbon, 2 from hydrogen, and 1 from oxygen. Basically, the H and O will make up the water molecule. So you will need a water molecule and a carbon atom. This is an important thing that you need to know as well. Hydrate means water, hence why we need water to make carbohydrates. So there are three types of carbohydrates: monosaccharide, disaccharide, and polysaccharide. So I will go through monosaccharide first. Monosaccharide are simple sugar and a sugar which makes up the simple sugar molecule. So the example is treosis, pentosis, and hexosis. As you can see, the carbon atom for each is 3, and for pentose is 5, and for hexose is 6. Basically, we need 3 carbon atom to make up a triose, 5 carbon atom to make up a pentose, and 6 carbon atom to make up a hexose. So one thing that I can explain here is that the general formula for monosaccharide are C, H2, O, and in the bracket we have N. So basically N is the number of carbon atom. So example, they would like to know the general formula for hexose. So it will be CH2O and 6. So then you can also times it inside depending on the question. So this is basically where you need to put the carbon atom number. Next is an example of hexose is glucose, fructose, and galactose, and for pentose is ribose and deoxyribose. It's okay if you just have to know the trioses, pentoses, and hexoses. That is more than enough. Next is pentose and hexose have more carbon atom that they can form a ring from the hydroxyl group. This hyphen OH means hydroxyl group on one end to carbon on the other end. So they have pentoses and hexoses have 5 carbon and 6 carbon. So they can actually form a ring when hydroxyl group from one end and carbon on the other end combines. Next is joining of hydroxyl group and a carbon. So if the join is above the ring, we call them as beta glucose. If it's below the ring, we call them as alpha glucose. I'll give you an example of alpha and beta glucose here. So this is an example of monosaccharide. As you can see, one is below and one is above. So the one that is below is alpha glucose, the one that is above is beta glucose. So one way that you can remember this is that this is above and this is below. So A is always next to B. So if you can remember one of this, you can remember the vice versa of this. Next is the function of monosaccharide. The function of monosaccharide is that it is a source of energy for respiration and it's a building block for larger molecules. Next, we will look into disaccharide. Disaccharide is the joining of two monosaccharide that are linked by condensation. So this is very important. Glucose and glucose will make up maltose at 1,4 glycosidic bond. Glucose and fructose will make up sucrose at 1,2 glycosidic bond. Glucose and galactose will make up lactose at 1,4. So basically the disaccharide are actually maltose, sucrose and lactose. Next is that how they form. So one hydroxyl group from one ring and hydrogen from another ring will make up the water molecule, which allows the production of oxygen bridge to form a disaccharide. So this is an example of formation of a disaccharide. So as you can see here, OH from one end and H from another end will form water, which will then remove. So removal of water is condensation. So once it undergoes condensation, it will form a glycosylic bond. You can see them here. Here is already a H and here is already here is already a C and here is already a C. The one that is left out is O. Hence COC. This is the glycosidic bond. Alright. So next is polysaccharide. Polysaccharide are large and complex joining of many monomers with the help of glycosidic bond. Polysaccharide must be formed as glucose as it is very reactive, hence polysaccharide are more in it. So whatever that is in disaccharides such as glucose and stuff, they have to be formed as polysaccharide, which is the end product of carbohydrates, because glucose is very reactive. Polysaccharide are the most inert one, hence it's not reactive. Once they undergo processes that's for the body requirement, that's when it will form back disaccharide and monosaccharide. So the end product is starch, glycogen and cellulose. So how do these form is by amylose and amylopectin will form starch. 1,4 linked alpha glucose with 1,4 linkages will form glycogen, and the polymerization of 1,4 beta glucose will form cellulose. Alright, the joining of 1,6 linkages from a 1,4 linkages for cellulose. In order to form a 1,4 link, the adjacent ring must be rotated to 180 degrees. So it cannot be just 1,4, 1,4 like that, right? because then you can't form the rotated, hence why they say that for cellulose, one four link in the adjacent ring must be rotated. So starch is found in the chloroplast, glycogen is found in the animal cell for storage of carbohydrates, and cellulose makes up the plant cell wall. So starch and glycogen are the same thing. However, starch is just solely found in chloroplast and glycogen is found in animal cell. Next is the biochemical test that you need to know. So the biochemical test that involves the testing of carbohydrates is a Benedict's test for reducing agent, for reducing sugars, negative Benedict's test for non-reducing sugar, and iodine test for starch. So the summary and the procedure of the test will be informed later. Alright, so one common thing that we always see in the forming of molecules is dipoles and hydrogen bond. So dipoles and hydrogen bonds are basically covalent bonds that have a shared pair of electrons. However, the unequal distribution of charges are called dipoles. So you can see this here. This is an example of, this is a structure of the water molecule. So we have the oxygen atom and the bond that form and the hydrogen atom. However, the formation of water molecule are known as dipole, which is the unequal distribution. So see, hydrogen carries the partial positive and another hydrogen carries a partial positive. Oxygen carries the partial negative. Hence why they are able to join with each other. Next, we're going to be looking into lipids. There are 4 types of lipids here. So the first is fatty acid. We have fatty acid, alcohol and ester, triglyceride, and phospholipid. So fatty acids are actually acidic group of carboxyl group. COOH are carboxyl group. And fatty acids are known as unsaturated lipids. So for alcohol and esters, the hydroxy group are attached to the carbon atom are called alcohols. Acid and alcohol forms an ester by the ester bond or ester linkage. So one way that I can explain this is that alcohols are basically whatever that has OH at the back, and ester have COO at the back of their general formula. As long as you can see these two together, you can identify them as ester and alcohol. Next is triglyceride. Triglyceride are fats and oil, and to form a glyceride you would first need an ester plus a fatty acid to form a glycerol. So we first need ester and fatty acid to form glyceride, and then to form a triglyceride we need a glycerol and we need 3 fatty acid to form triglyceride. The reason why it's tri is because we have 3 fatty acids. So triglycerides are insoluble in water, as the 3 hydrophobic tails here, the fatty acid are hydrophobic tails. So the 3 hydrophobic tails are actually, they will repel with water, hence why they are insoluble. And yeah, so the role of triglyceride is that they are energy reserves, they act as insulator, and they are the metabolic source of water. Next is the phospholipids. Phospholipids have two phosphate head and two hydrophobic tail. Phospholipids are soluble in water. The reason why they are soluble in water, even though they have the hydrophobic tail, is because, as you can see here in triglyceride, three fatty acid, three hydrophobic fatty acid tails will be insoluble. Only three can be insoluble. However, for if you have two fatty acid, one out of three of the fatty acid will become the phosphate head. Hence why they are actually soluble in water. So the biochemical test needed to test lipids is called an emulsion test. Next, we will look into protein. Protein are actually amino acids that are joined by peptide bond to form proteins. So what is a peptide bond? Peptide bonds is formed by OH from COH and H from the amine joins C and N and to form the water. Alright, so basically this is the formation of peptide bond. So we have two amino acids here. So from the COOH, we will need OH, and from the amine group we will have H to remove as water so that they can form. So they will undergo condensation, and once it has been removed as water, you can see the formation with C and N. That is what we call a peptide bond. So the types of protein is that they have from primary up to quaternary structure. So for primary structure, they are actually the particular amino acids contained in a chain, and the sequence in which they are joined are known as polypeptide. So primary structure are polypeptides, basically long chain of amino acid. And after primary structure, they would form a secondary structure. Secondary structure is basically the polypeptide may coil to form alpha helix chain, or they will be folded into beta pleated chain, beta pleated sheet. So this alpha helix and beta pleated are joined together by hydrogen bond, which is the CO and BNH. After forming a secondary structure, they will form a tertiary structure. Tertiary structure is a compact structure of protein molecule resulting from 3D coiling of the already folded chain. So this is, whatever that is formed in secondary structure, it's all in one long chain. However, for tertiary structure, they will become a 3D coiling. So there are four types of bond for forming the tertiary structure. First is hydrogen bond, which is between R groups, disulfide bonds, which is between two molecules, ionic bonds, which is between R group and an amine and carboxyl group, weak hydrophobic reactions, which is between R group and nonpolar or hydrophobic. Lastly, this tertiary structure will form a quaternary structure, which is a 3D arrangement of the association of different polymer chains held by the same four types of bond, which is this one. So amino acid. So basically, they will have a C in the middle, a hydrogen, amine group, carboxyl group, and the R. This is the side chain. This R chain can be anything based on the amino acid codons that are available. So next is the function of protein. Basically, the function of protein is that they act as enzyme, they are the component of cell membrane and their functions. They are hormones, they carry hemoglobin and myoglobin to carry oxygen. They are antibodies to attack pathogens. They are also collagen to add strength to tissues. They exist in our hair, nails, and skin as keratin. They have actin and myosin for muscle contraction, and they are storage of products. We have two types of protein, which are globular and fibrous. So these fibrous protein are long and parallel chain. They have cross link between molecules. They are insoluble in water. They are stable. They have mechanical and structural support, and they have keratin and collagen. For globular, they are polypeptide folded into compact spherical shape. The hydrophobic are inwards, hydrophilic are outwards. They are soluble in water. They are unstable. Metabolic reaction takes place, and the examples are enzyme, insulin, hormones, and antibody. So this is basically the um, differentiation between fibrous and globular protein. The biochemical text involved is a Burette test. Last but not least is water. Water, these are basically the function of water. Why do we need water? So first they act as solvent. So water are solvent because the water are attracted to ions and polar molecules, which then will separate as which then will separates the water molecule. So the separation of water molecules allows them to bind with the solution. So this is how they bind. So if the solution is positively charged, the oxygen will be facing the positively charged. However, if it's negatively charged, the hydrogen will be closer to the solution. So if it's positively charged, the oxygen will be attracted. If it's negatively charged, the hydrogen will be attracted. Water acts as a transport medium. So transport medium, usually it plays a role in the organ system and vascular tissue. The mechanism of solvent actually works in the transport medium as well. So for high specificity to heat capacity, for water, it is 1 kg to increase 1 degree Celsius. Hence, the specificity is very high, hence lower energy is required. This will result in no fluctuation of water. High latent of heat vaporization. Basically, the water molecules stick together with strong hydrogen bond. Hence, it's very hard to be broken up into gas. That's why they have high latent heat of vaporization. For density and freezing point, the freezing below 4 degrees Celsius. The limit for water is 4 degrees Celsius. So below 4 degrees Celsius, the density of the water will start to decrease. That's why ice will float on liquid. This will help to maintain the circulation of nutrients in ocean. Next is high surface tension and cohesion. Cohesion is the water molecule that stick to each other, which increases surface tension. This will allow small organisms to travel on water. Last but not least is reagent. Water as reagent usually occurs in photosynthesis or hydrolysis. So in photosynthesis, the energy from sunlight will be used to separate hydrogen and oxygen in water. And hydrolysis is the breakdown of larger molecules into smaller molecules.